Hello and welcome back. This video we're going to kind of review a little bit about what happens in oxidative phosphorylation with the electron transport chain and chemiosmosis. This video I'm also going to talk about where things can go wrong and what happens if oxidative phosphorylation doesn't happen the way it's supposed to. Okay? So I've redrawn my diagram up here. Okay? This side of the membrane, this would be in the matrix of the mitochondria. We've got our intramembranous space here. This is our outer membrane of the mitochondria. All right. Remember, we generated that hydrogen gradient because we were taking electrons from NADH. Those are deposited into the electron transport chain. Those electrons bounce around in the membrane. They exit the membrane as a low energy electron. Remember, they start as high energy, end as low energy. That's what allows the hydrogen to move from the matrix of the mitochondria into the intramembranous space. At the end, we've got our electron plus hydrogen plus oxygen, okay? And that's going to give us our waste product out of this, our water, okay? So we've actually got, in this case, you'd need 4 hydrogen and you'd make 2 water out of this, okay? So water is one of our waste products associated with cellular respiration. It happens at the end of the electron transport chain there. And remember, oxygen is our final electron acceptor. In the process of oxidative phosphorylation in the electron transport chain, okay? So oxygen is what makes this aerobic respiration. This is where the oxygen is needed in cellular respiration, okay? The step of cellular respiration where it's needed is at the end of the electron transport chain there, pulls the electron out of the electron transport chain, all right? The other part of oxidative phosphorylation is going to be chemiosmosis, okay? And that chemiosmosis is going to happen through ATP synthase. So remember, of the proteins that are in the mitochondrial membrane, this is the one I want you to know the name of, is ATP synthase, okay? With that ATP synthase, what happens? Those hydrogens that are in the intramembranous space are at a high concentration. And remember, when we're talking about diffusion, diffusion goes from an area of high to low. And that's what we've got here. And the way those hydrogens move back across the membrane is through ATP synthase. So that hydrogen shoots through ATP synthase because it wants to diffuse back to an area of low concentration. And what we have happen there at ATP synthase is our ADP and a free phosphate are going to then be reconnected to generate our ATP, okay? So the ATP that's generated in oxidative phosphorylation is going to be generated during chemiosmosis. All right? And this is going to be where we have the bulk of ATP made in cellular respiration, is going to be through chemiosmosis. All right? So that's how we generate the ATP. Now, when things go wrong, you're going to muck with this system, okay? If our NADH can't put the electrons into the electron transport chain, first thing that happens, we no longer generate NAD, okay? And we're not going to generate, let me get an actual eraser here instead of my fingers. We're not going to generate the hydrogen gradient. Okay? So if we don't generate the hydrogen gradient, this step isn't going to happen. We aren't going to get chemiosmosis. We aren't going to get those hydrogens coming back across. We aren't going to get ADP in our phosphate converted to ATP. So if we don't get electrons into the electron transport chain, chemiosmosis can't happen because we need that hydrogen gradient, okay? So that's one way where we can have things go wrong, okay? Another way we can have things go wrong, maybe we get NAD going in, okay? But our membrane ends up being a little bit leaky, all right? If our membrane is leaky, our hydrogen can come back across, it can reach equilibrium. Again, if we don't have that hydrogen concentration gradient, we don't get chemiosmosis to happen, okay? So that process of inhibiting electrons going into the electron transport chain, there are various chemicals that can block that process. Some of the chemicals used in a lot of flea dips and flea shampoos, this is the part of cellular respiration that's actually being blocked. That's why those particular shampoos or dips say you shouldn't ingest them, because if you take them in, you're going to stop cellular respiration inside your body. Applying it to the outside, what you're wanting to do is you're stopping cellular respiration in those fleas, all right, which is what's going to lead to their death. So there are going to be chemicals that are going to block here. There are chemicals that make the membrane a little bit leaky. Things like cyanide, and there's a diet drug, I think it's called DNTP or DNHTP, that essentially have that process. They make the membrane a little bit leaky. You don't generate that hydrogen gradient, and you don't get chemiosmosis to happen. And that's the important step of oxidative phosphorylation, okay? There are other drugs that will block the ATP synthase, okay? But the point of that is if you block any point in this process during the electron transport chain, it'll interfere with chemiosmosis. If you interfere with chemiosmosis, you're not getting that bulk of ATP made, okay? So that's how various agents, various toxins and poisons work. It inhibits cellular respiration, and that process is important, an important day-to-day function for the cell, because that cell has to generate ATP, okay? Hopefully that made sense, okay? If you have any questions, please let me know.